To make the DNA spots on the slides, we use a pH indicator solution, and we use rapidograph pens for the equal sized spots for all of the DNA samples. The pens are filled with pH indicator solution, phenolphthalein and thymolphthalein. Phenolphthalein is used for the red color dot, thymolphthalein for the blue color dot, and the solutions are made in a 10 milligrams per milliliter of ethanol. The pens are filled with the solution, and we have the 3 different colors. We have red for one condition, blue for another condition, and purple for both conditions. So for example, in our breast cancer microarray, the breast cancer tissue would, if only a gene is expressed in breast cancer tissue, it might be red. If it's only expressed in non-cancer tissue, it might be blue, and if it's expressed in both tissues, then it would be purple. The purple is a 1-to-1 mixture, and when we fill the pen, we take a little bit out of a tube that we've pre-mixed with the solution, put it into the reservoir for the rapidograph. This is where the ink would normally go, and then screw it onto the pen tip. Put that into the holder, and your pen is ready to go. You always want to check it to make sure it's actually working before you actually use it on your slide. These dots that will be created will be equal sized and have an equal amount of pH indicator in it. At neutral pH, or the pH of air at this point, as well as water, the spot will look clear. Okay, so now the slide will be positioned with a template, and let me show you a closer view of the template. This is an acrylic template that has holes, 50 holes placed into it. This is where our pH indicator or DNA spots will be made. Okay. And then taking the rapidograph pen, poke it into each of the holes to make the different DNA spots. On this particular slide, there are 6 spots that will be red, 5 spots that will be blue, and the remaining 39 spots will be purple. So in this condition, 6 pieces of DNA will be expressed only in breast cancer tissue, 5 spots will be expressed only in non-cancer tissue, and the other 39 spots are expressed in both tissues. These numbers can obviously change depending on what your conditions are, what your genes are, and what your scenario is actually trying to express and show. And once your slide has been completed, the slide can be saved up to 2 weeks at room temperature and several months in the refrigerator. And there There is your slide with spots.